Good afternoon. Welcome back to winter. Right? We had spring and all of a sudden it's cold again. Welcome back to Virology. Today we will talk about persistent infections, the kind that last your lifetime. You all have these viruses, or most of them, that we're going to talk about today, including me. So last time we talked about acute infections, rapid and self-limiting. In contrast, a persistent infection is long-term, the life of the host. So HIV/AIDS, for example, is a persistent infection. We will talk about that separately. The characteristics of the persistent infection are similar for certain viruses, so herpes simplex viruses, it's similar, and human cytomegalovirus, which is another herpes virus, is different, and so forth. And most persistent infections begin as an acute infection, an infection of defined duration, sometimes with disease, and then it's not cleared, and you have a persistent infection. So some of the reasons why you get persistent infections. The primary infection, the acute infection, is not cleared by the immune response. We'll talk about ways that happens. In many persistent infections, virus particles, viral proteins, viral genomes continue to be produced, but not all. You'll see exceptions. So there are some cases where there are just genomes and no protein, and there's some cases where there are genomes and a few proteins, but no virus particles. And there are cases where there are infectious virus particles made. So the whole spectrum can happen. There's not a single mechanism that would apply to all viruses, but in general, viruses that don't kill cells, remember, viruses that don't cause cytopathic effects, when that happens and immune defenses are reduced, remember, a cytopathic virus gives you a good immune defense. A non-cytopathic virus doesn't, and it's more likely to persist. And all of these viruses undergo immune modulation, and that certainly contributes to persistence. These are some of the human persistent viral infections. The ones with the red asterisk we're going to talk about today, but you can see there's both DNA and RNA viruses. They persist at many different sites, as you can see in the middle column. And the consequences are quite varied, as you can see. We'll talk about that. One adenovirus persists in adenoids, tonsils, and lymphocytes. We don't know the consequence of that at all. An important part of clearing a virus infection, I hope you remember this, is the cytotoxic T lymphocyte response. Remember, we have a virus-infected cell. These cells have MHC1 on their surface, right? And they have peptides displayed in MHC1, both cell peptides and virus peptides. And cytotoxic T lymphocytes have a T cell receptor which they query the peptides with. And if the peptide fits in the T cell receptor, that means it's foreign, and then the CTL will kill the virus-infected cell by inducing apoptosis. We talked about this before. This MHC1 system, where most cells in your body display peptides in the context of MHC1, is subject to quite a bit of virus modulation. Now, the faded picture here we showed you before, and that is how peptides are displayed in MHC1. And then in more sharp focus are the ways that viral proteins antagonize MHC1 function so that their infected cells are not destroyed, right, by CTLs. So part of the CTL part of the presentation response is to take viral peptides. So this is an infected cell now, and there are proteins, they're viral proteins as well as cellular. They're put through the proteasome, which is a big protease, chops it up into peptides. The peptides are transported through a transporter into the ER. And we already have two sites of antagonism here. So ABNA1 is antagonizing peptide generation. And these two viral proteins, notice all herpes viruses, we're going to talk about them a lot today, they are inhibiting the transporter. So the peptide never gets to the MHC1. Once the peptide is in MHC1, it's transported to the cell surface. And there are viral proteins that interfere with transport. And then even if the MHC1 is on the cell surface with a peptide in it, there are viral proteins that will remove it from the cell surface and send it to the lysosome. They basically induce endocytosis of the MHC1 and send it to the lysosome where it's degraded. And here we have some other interfering. Here's MHC class 1 being sent to the proteasome by a viral protein here. And some viral proteins even inhibit transcription of MHC class 1 genes. So all of these viruses cause persistent infections. And this is one of the reasons why, because they're inhibiting MHC1 presentation of peptides. And that prevents the infected cell from being killed. It's modulation of MHC1. Now, many viruses do something called CTL escape. Remember, a CTL is recognizing a peptide, a viral peptide in the context of MHC1, right? Here it is, right there in this picture on the right. Now, it's persistently infected hosts. Virus is reproducing for a long time, so there's plenty of time to produce mutants. And so proteins are made with amino acid changes in them that are not recognized properly by MHC1. So here is MHC1. Actually, it's MHC2, but it could be 1 as well. MHC2 is in the antigen-presenting cell. And this is a nice peptide fitting in MHC2. But in the course of virus replication, amino acid changes occur. These blue change here. So now the peptide doesn't fit into the MHC2. And often these amino acid changes also prevent the T cell receptor from recognizing the peptide in the context of MHC1 or 2. All right, so these are CTL escape mutants because the peptide is no longer recognized as foreign. So herpes simplex virus does this. Hepatitis C virus does this. It's in a way like antibody escape. Remember, antibodies neutralize viruses, and viruses can vary to escape neutralization. But in contrast, CTL escape does not facilitate population spread, whereas antibody escape does. If you're infected with influenza virus and that virus varies in you so that it escapes antibodies, you can spread that virus to someone else and it will escape their antibodies because antibodies work the same way in everyone. But MHC is polymorphic. Your MHC is not mine. So a peptide change that escapes my MHC is not likely to have any effect on anyone else. So they don't facilitate spread. What they do is facilitate the infection keeping going in a particular individual, okay? So that's a really important concept, that these MHC escape mutants only facilitate reproduction in the host and not helping spread to another host. Now, when T cells are activated, they become CTLs in part. Some of them become CTLs and kill virus-infected cells. We've just told that. Sometimes the infected cell will kill the CTL instead of the CTL killing the cell. Okay? the CTL may die instead of the target. So this is an example of a viral antagonism. And it is done by receptors on the surface of the cell. 3 different receptors shown here: TNF, FAS-L, and TRAIL receptors. And binding of ligands to these receptors induces apoptosis. This is a normal process to limit immunopathology. So you don't want to have T cells, CTLs around all the time, right? So once they're induced and activated and the infection is under control, they're gotten rid of by apoptosis. But some viruses take advantage of that. For example, HIV and cytomegalovirus induce Fas ligand on the infected cell surface. So Fas ligand is the ligand for this receptor that induces apoptosis. So what happens is the T cell engages the infected cell, the Fas ligand engages Fas ligand receptor on the CTL, and the CTL undergoes apoptosis before it can kill the infected cell. Okay, so it's another way of antagonizing T cells. So there are ways of antagonizing that are going to play into what makes a virus persist. Now, many tissues have what we call reduced immune surveillance. You know, the immune system is not the same everywhere. Some places can't have a rigorous immune response because it would be damaging. For example, the central nervous system, right? It's in a skull. If you get inflammation and it starts to swell, there's nowhere to go. That's bad, okay? So there's reduced inflammation in the CNS. And when viruses get in and cause inflammation, this is very worrisome. And they have to treat you with steroids, for example, to reduce brain swelling. CNS, the eye, right? It's an encased sphere with fluid in it. If you get inflammation, it's not good because it can't expand very much. Skin is another one. They're devoid of initiators and effectors of the immune response, in particular the eye contains high levels of Fas ligand, so that if any T cells should get in, they'll be killed immediately. So this is to protect these tissues from immunopathology, but unfortunately, the result is you get persistent infections because you can't clear the virus from them. And here's an example of one that was reported back. Well, in 2015, there was a huge Ebola virus outbreak in West Africa. And after it was all over, there was this one patient here, but there were others. Among the survivors of Ebola, complications that include uveitis. Do you know what uveitis is? Inflammation of the eye. So this is the first time this was seen after the Ebola is cleared. So all these patients are PCR negative, like 30, 60 days after infection. And then suddenly they're getting uveitis. And this is a case report of one patient. Here's the patient, Ian Crozier. He was a doctor who went to West Africa to take care of patients. He got Ebola. When he came back to the US, they treated him, he survived. Then like 2 months later, one of his eyes changed color. What would you do if your eye changed color? Would you just ignore it and keep studying? He went to the doctor, which is unusual because he is a doctor, and usually doctors don't go to see doctors. He went, and he was an Ebola survivor, so they found Ebola virus in his eye, in his vitreous humor, infectious Ebola virus. It wasn't shedding, so he was not a danger to anyone, but that was causing his eye to change color. So what happened is somehow the virus went into his eye, and it was never cleared because there's a reduced immune response in the eye, right? And then probably reproduced very slowly and caused the eye change. So I mean, you guys don't have Ebola, but if you ever go somewhere and you get Ebola and your eye color changes, that's why. So that was the first time we noticed that Ebola virus could persist, because we thought it's an acute virus infection, but it persists because it goes in the eye and it's not cleared, right? Normally, if it's elsewhere, it's going to be cleared. In 2021, 6 years after the 2015 outbreak, some of the people who survived, men, transmitted infections to women sexually. It turns out the virus persisted in their testes. That's another immunoprivileged site. And it probably reproduced very slowly. They didn't have any symptoms, and they passed it on to others. So turns out that many hundreds of men had Ebola virus persisting in seminal fluid. I just want to show you the genomics of this outbreak. And so there's a phylogenetic tree of genome sequences of Ebola virus genomes. These are the genomes from 2015. And in 2021, the little outbreak caused by men passing it sexually made this little branch on the tree down here. And that's expanded on the right here. There were 4 cases, I think, where they passed it on. And obviously, this virus is just related to what was circulating 6 years before, because they harbored it pretty much there were no base changes, so it wasn't even replicating in them. So anyway, some acute virus, this is a good illustration of how the immune system plays a big role in persistence. Okay, some viruses infect immune cells. Measles virus infects lymphocytes, and as we said, in antigen-presenting cells. HIV infects CD4 cells, monocytes, macrophages, and dendritic cells. So if you infect cells of the immune system and destroy them, obviously the immune system is now defective and you get a persistent infection. Okay, so the immune system interaction with these viruses plays a big role, and even an acute virus can suddenly become persistent if it goes into the wrong place. All right, first question is, which of the following are features of persistent infections? They last the lifetime of the host. Viral immune modulation is involved. Immune cells may be infected. They may occur in areas of reduced immune surveillance. All of the above. Well, it looks like we're done. Yeah, you go 13 out of 13. It is all of the above. That's right. Everything is involved, which are features of persistent viral immune modulation, and immune cells will be infected, are part of that. All right, I want to go through some persistent virus infections and talk about how it works. This first one is measles virus, which you may remember we talked about last time as an acute virus infection, right? Paramyxovirus, extremely contagious. 2023, 663,000 global deaths, totally preventable with vaccination. You get lifelong immunity after infection. Classic acute virus infection. So where's the persistence? There's a disease called SSPE, subacute sclerosing panencephalitis. This is a fatal neurological disease, 100% fatal. So children get measles, they recover, they're going on with their lives. 10 years later, 5 to 10 years later, they start having cognitive issues, they start having motor issues, and eventually they die. There is 100% fatality. And they have measles virus in their brain. It is not infectious virus. It is a defective particle, but it's able to spread between neurons and cause damage. And in particular, you may remember the measles virus has 2 glycoproteins on the surface, the H and the F. 2 glycoproteins on the surface, the H and the F, and the F has changed, has amino acid changes that allow receptor-independent cell fusion. Now, why this happens, it's not clear. Maybe in a so we know that 1 in 1,000 kids get encephalitis right after measles, right after the measles infection. So it could be that there's virus in more kids' brains and it stays there. It's not cleared because it's an immunoprivileged site. and it undergoes, the genomes mutate, and eventually one arises with these F protein changes that allow fusion. So it's the fusion between cells in a receptor-independent manner that damages the brain. Now, with the elimination of measles in 2000 in the US, we eliminated SSPE. But now, since measles cases are coming back, we'll soon see a resurgence. It's going to take 5 to 10 years for us to see this. This is completely preventable by vaccination. Look at it, 1 in 10,000. It's not trivial. This is not a nothing disease, as so many people portray it. They're lying when they say that. If your kid gets SSPE, you're very upset. And it's preventable by vaccination, so there's no reason to go through this. Okay, polyomaviruses. Remember SV40? We talked about quite a bit. We're going to come back to it, actually, next week. The polyomavirus family is big. There's Wookiee polyomaviruses, there's AV polyomaviruses, and there's ortho. And the ones with the red asterisk infect people. And so here in the Wookiee polyomaviruses, all of them infect people. The name, by the way, comes from 2 isolates, one from the Karolinska Institute, KI, and one from Washington University. That's where Wookiee comes from. It wasn't actually isolated from a Wookiee, although I'm happy to think it was. All right, the Wookiee would have been fine because these are not causing disease. And down here, these are bird polyomaviruses on the upper right. Down at the bottom are some others that infect various species, bats, mice, et cetera. Here's SV40, but then BK, JC, TSP, and MCP. These are all polyomaviruses that infect humans. Now, most people are infected with some variant of one of these polyomaviruses. You get it early in life, and your human populations are highly seropositive for these various polyomaviruses. They infect a variety of organs, kidney, intestine, respiratory tract. Some people shed 100,000 particles per mil in urine. Okay? Remember, when you flush the toilet, you make an aerosol. 100,000 particles per mil. We don't know why they persist. They persist in lots of organs without causing damage. They must be in some kind of stasis with the immune system. But there's one case where they can cause damage, and that's the disease called PML, progressive multifocal leukoencephalopathy. It's a brain disease. And what happens is, if you have multiple sclerosis, you may be given a monoclonal antibody called Tisabri, which kills B cells, because B cells are part of the pathogenesis of MS. And so they wipe out your B cells, you don't have MS, but your polyomaviruses start to replicate, and they replicate in your brain. They cause a potentially fatal brain disease. And so at that point, you have to make a decision about whether to stop your Tisabri or not. So you suppress the immune system, and these viruses come up. So you can see they're persistent because there's some kind of arrangement with the immune system. If you want to learn more, I did a podcast a while ago with a physician who works on these viruses. Okay, anelloviruses. Again, over 90% of humans have these viruses. They are DNA viruses, icosahedral DNA viruses, with a single-stranded circular genome of 3 to 4 kb. They're in all of you probably, or at least 90% of you. They are transmitted by saliva, mother to child. They are transmitted sexually. They are transmitted fecal-orally through blood transfusions. You know, when you give blood, they don't check it for anelloviruses, but they're there because anelloviruses don't cause disease. If they threw out every pint of blood that was anellovirus positive, we wouldn't have a blood supply because we use donor blood. We don't know how to make blood yet. So these anelloviruses are benign, but when you get immunosuppressed, the levels go up. Again, signifying this, the immune system keeping this in check. By the way, they infect many tissues. You can find it in blood, saliva, urine, liver, and kidney. So again, everybody has these. These are examples of persistent infections that are benign. They don't hurt you unless you are immunosuppressed. But we'll get into some now that cause damage. And the first one is hepatitis B virus. We've encountered this virus before, right? It's the DNA virus with the double-stranded genome that has a gap, and it has a protein linked to it and a piece of RNA, and has reverse transcriptase as part of its reproduction cycle. This is transmitted among humans by exposure to blood because the virus has a viremic phase. This happens at childbirth, during transfusions, sex, drug use, tattooing, nosocomial, in the hospital. So childbirth, many mothers are Hep B positive. And so their child is going to acquire Hep B. And even if the mother is not positive, the child is going to encounter Hep B early in life. So we vaccinate newborns right away. And you'll see why in a moment, because, well, it's right on this slide. Only 5 to 10% of newborns resolve the acute infection. They go on to be persistently infected their entire life, and they can develop liver cancer as a consequence. On the other hand, if you're infected as an adult, 95% of adults resolve infection. And the main target is the hepatocyte. That's hepatitis B virus. Now, I tell you this because several anti-vaxxers have said that babies should not be vaccinated for Hep B because it's a sexually transmitted infection. Why should babies be infected? So either they're lying or they're giving you misinformation because many babies are infected at birth. And vaccination has made a huge decrease in the amount of liver cancer that these individuals develop. So remember that when next time you hear this, please, because many people don't know, right? People just don't know what facts are. Someone says, you don't need to vaccinate them. Okay, I don't know anything. So you need to vaccinate kids, no question about it. Of course, also if you work in a lab with human cells, which could be contaminated with virus, you also need to be vaccinated as well. If you're a healthcare worker, you have to be vaccinated. Here are the numbers. Very hard to get up-to-date numbers. This is from 2022. 254 million people are currently infected globally. These are the countries where most of them are. And you get, in 2022, we had 1.2 million new infections. There are currently over 6 million children infected less than 5 years of age. They're going to go on to get liver cancer. And 1.1 million deaths a year. This is a big problem, but we do have a vaccine, which we'll talk about later. Now, this is the pathogenesis of acute and chronic hepatitis. Chronic, acute on the left. So we get exposed at week 0, and these are weeks post-exposure. Then we're measuring a lot of different things here, okay? Let's look at DNA. The green, so this is a good measure of virus reproduction. It's peaking around here, and you can see that's when you get your symptoms. And these other names, HBs antigen, these are viral proteins. So one of the viral proteins, the surface protein, the spike, peaks very early, as you can see. And then you have antibodies made to the virus. This black peak here is alanine transaminase. That's a liver enzyme. It's not supposed to be in the blood, but it is because the liver is getting damaged. However, after so many weeks, you recover and the virus is gone. So that's really good. However, it doesn't always resolve. In 5% of adults, it does, but most children less than a year of age becomes chronic. The same DNA replication, but it never goes away. See, here in the acute, DNA went down after a year. Here, it's persisting. ALT, liver damage persists, so your liver is always being damaged. And despite having antibodies to viral proteins, the infection persists. You just have symptoms for that defined period, but the virus is reproducing in your liver, it's damaging liver, and eventually you get liver cancer. Interesting, I just said the virus is damaging. It's not, it's actually CTLs that are damaging the liver. The immune system is damaging liver cells. And what happens is many liver cells are damaged, they regenerate, they're damaged again, and after so many cycles of that, the T cells get exhausted and they're dysfunctional, so the virus is no longer eliminated. In the meanwhile, your liver is getting scarred. We call that fibrosis. You get liver failure, and eventually, after 20 to 30 years of these cycles, it's often asymptomatic. You don't have any symptoms, although if you look at people's eyes, they're yellow, because that's the jaundice from the liver damage. They will develop liver cancer, which is fatal. Okay, so this is vaccine preventable. Then we have hepatitis C virus. We have hepatitis A, B, C, D, and E. I think it went further. But hepatitis C virus is an RNA virus, a plus-stranded RNA virus of the Flaviviridae. It's enveloped. This is transmitted very much like Hep B, sex, drug use, tattooing, during birth, contaminated blood. We don't have a vaccine for this one. We should, so no one's complaining about vaccination. This is the global burden. Once again, 71 million in 2022. Actually, it's 50 million. 71 million was an older number, so it's slightly going down. 3.2 million adolescents, 1 million new infections a year, 242,000 deaths. So slightly less burden than Hep B. Here's the pathogenesis. Acute infection, you get infected by the roots that we just talked about. Elevated liver enzymes. So let's look at the graph here, that's better. We have Hep C RNA. On the left Y and on the right Y, we have the liver enzyme ALT. And then we'll look at acute infections, the red. So you have an early peak, these are months, an early peak of virus RNA that goes down. And then we have ALT, which goes up and then goes down. So it's an acute resolved infection, but in many people it persists. So you can see the blue is viral RNA in the persistent patients. The ALT rises and falls over the months of infection. So acute infection, 3 to 6 months, 10 to 40% of the patients recover. 60 to 90% go on to have a persistent infection. And then that continues for 30 years. 70 to 98% of patients have an asymptomatic persistent infection, but 2 to 30% have end-stage liver disease. Right? So again, the persistence is the problem here. This virus also evades immunity in many different ways, which explains the persistence. Now in the US, the opioid crisis has led to an increase in Hep C infections. You know that opioids are injected drugs that are increasingly being used. If you're sharing needles, you will share other things. And so the number of cases of Hep C in the US went from 16,000 to 41,000 in just a few years. These are the opioids, right? Heroin, fentanyl, opioids, methamphetamines, cocaines, all injected, which are shared features of persistent infections with polyomavirus, HBV, and HCV. A, genomes are present but not expressed. B, liver damage. C, kidney damage. D, virus particles are produced. E, all of the above. Okay, how did we do? Oh my gosh, this is all over the shop. The right answer is virus particles are produced. Polyomavirus, HBV, HCV. That's what they have in common, virus particles. Genomes are present but not expressed. That can't be if they're making virus particles, right? Liver damage. No, polyomaviruses are not causing liver damage. Nobody picked kidney damage. It's good. Okay, do you understand that? All right, so we're going to now talk about herpes viruses, different kinds of herpes viruses. And on this chart, we have our acute infection. Remember, the red is disease, the blue is virus production. So acute infection, defined period of virus production and disease. And then different kinds of persistent infections. Like the second one is herpes simplex, where we have, it's called latent, but it's a persistent infection because you have periods of virus production and disease, and then no virus is detectable. And sometimes you have virus with no disease, as you can see here. Then we have persistent asymptomatic. We've already talked about the polyomaviruses have this. You make virus your whole life without any disease. I just call them all persistent, but this is the textbook here. And then persistent pathogenic, which I don't think is right because these red bars are pathogenic, but this is, you have an initial acute infection, you have virus persistence, and then so many years later, death. So this is HIV, HTLV, and SSPE caused by measles. All right, let's explore these one at a time. So in these latent infections, so the reason I'm calling them latent is because in some cases, there are periods when no virus is produced, but the genomes are obviously still there. Sometimes proteins are present. The cells that have these genomes are poorly recognized, but the genomes persist so that you can make more virus and spread to a new host. If a virus went in a host and just persistently infect that host, it's a dead end if it didn't spread to another host. It's useless because the viruses need to transmit to other hosts, otherwise they're gone. Right? So the genome exists in different states depending on the latent infection, and we'll take a look at this in detail. For herpes simplex virus and varicella-zoster virus, it's non-replicating DNA in a non-replicating cell. A neuron does not replicate, so the viral DNA does not have to replicate. In some cases, Epstein-Barr, cytomegalovirus, human papillomavirus, HBV, Kaposi's sarcoma, herpes virus, they're not infecting neurons, they're infecting a dividing cell, so the viral DNA has to replicate. And there's one example, human herpes virus 6, where the viral genome integrates into the host chromosome, so it replicates with the host. We'll take a look at this. Herpes simplex virus first. In the US, over 80% of people are seropositive with genomes in the peripheral nervous system. The peripheral nervous system is outside of the central nervous system. The central is your brain and spinal cord. Anything outside it is peripheral. Many, many millions of Americans carry viral genomes in their peripheral system without any symptoms. 40 million Americans experience recurrent herpes disease in the form of cold sores. There are two herpes simplex viruses, simplex 1 and 2. 1 has a preference for oral infections. 2 has a preference for genital infections. But they can mix. It's not absolute. And this is a well-adapted pathogen because it infects many, many people and mostly causes minimal disease. So let's explore how this works. You get infected at a young age. Your parents are shedding herpes simplex virus at some point. It's in their saliva. They kiss you, and it comes into your mouth, your mucosal surface, and the virus is infecting epithelial cells. It reproduces in these cells, passes through them, and then enters nerves, typically sensory or sympathetic nerves, travels to the sensory neuron body, and there it becomes latent. So you may have a primary infection. It's either in a parent or you can have various symptoms. and then it becomes latent. And so in utero, you can be infected as well. Or during birth, 80% of babies are infected during birth. Okay, so the virus is now latent. This is a peripheral ganglia. It's a collection of neuron cell bodies. It's shown, one is shown there, but there are more than one. All right, so here's a schematic of what we just talked about. Here are your epithelial mucosal cells, and here's a nerve innervating them that goes to the local ganglia, lots of neurons in here. And there is the viral genome. It is silenced and coated with nucleosomes. Multiple copies of the DNA remains in the nucleus of the neuron. It does not need to replicate because neurons do not replicate, right? And there it is forever. Drugs and vaccines cannot cure a latent herpes infection. Unlike love, herpes is forever. Maybe one day we'll be able to cure it. But on this lecture, if you go to the YouTube page when it's posted, or the YouTube of this lecture for any previous year, you'll see people writing, Dr. So-and-so cured my herpes with this or that treatment. It's all a lie. You can't be cured. You either didn't have it or you're lying. But I have a moderator who takes them all out and they just keep coming back. I don't get it. I guess they want to promote Dr. So-and-so's phony treatment. Can't cure it at this point. Now, in the nucleus, the genome does not make proteins. It makes small RNAs called latency-associated RNAs, or LATs. Those are processed to microRNAs. No proteins are made from them. We think the microRNAs are involved in keeping the genome silenced. It is silenced at this point. No proteins, no RNAs except for LATs, no virus particles, of course. Okay, reactivation. At some point, this genome begins to replicate in the neuron. New virus particles are made. They travel to the mucosal surface, infect those cells, and they're shed, and that's how they appear. That's how they transmit. So small number of neurons reactivate. The virions appear in mucosal tissue innervated by these ganglia. Sometimes you get blisters on your lip or in your mouth or on your nose. Not always. And that's how it is transmitted. Sometimes you are shedding without a blister. You know, if you have a blister, you really shouldn't get intimate with anyone. But people do, but if you don't have a blister and you're shedding virus, not much you can do. So the immune response is too slow to prevent shedding. Eventually the immune response kicks in and stops the shedding, but it's too slow. There's antagonism going on, so that's why you get virus production. Now, some people reactivate every 2 to 3 weeks. They get cold sores. Some people never reactivate. This is one of the places where herpes simplex 1 viruses become latent, the trigeminal ganglion. There's one on either side of your head. It is where it's full of neurons that connect to the lips, the face, the nose, the forehead. And so when herpes is latent in it, well, remember, you get infected at a mucosal surface, the virus goes via the nerve to the trigeminal. It goes latent there. It's reactivated. It goes back down the nerve. You typically get cold sores on your mouth, but you can also get them on your face, your nose, and your forehead. And that's the anatomy of what's going on there. And for the genital herpes, you have ganglia down there that do a similar thing. So what causes reactivation? We understand this best for herpes viruses, so I'll tell you in some detail. So if you're latently infected, sunburn will reactivate. You go to the beach, a few days later, you have a cold sore. Physical or emotional stress, taking exams will give you cold sores. Sorry, I try and reduce it as much as possible. Nerve damage, hormonal imbalance, steroids, these are some of these stresses that cause reactivation. And we believe they stimulate the production of viral proteins that are needed to activate transcription. Now, I want to remind you of the transcriptional program, excuse me, of herpes viruses, shown down here. You have an immediate early promoter that has to be transcribed before early proteins can be transcribed, which are needed to replicate DNA and make late proteins, right? This IE promoter does not work in a cell. The cell proteins don't recognize it. So when the virus infects, it brings this protein VP16 along with the virus particle, which helps to get that promoter working. Now, when you're latently infected, there's no virus particles around, so there's no VP16. So for years, people could not figure out how reactivation worked, because you have to make IE proteins first before you can make virus particles. But here's the answer. A very interesting series of studies were done, which show that the herpes genome, so here's a neuron in a ganglia at the top, and there's a herpes genome, a circle, and that genome is wrapped around nucleosomes, right? And these nucleosomes are methylated to make them tight, and so transcription is shut off. So that's maintaining latency. When you get stressed, you activate protein kinases in your neurons. And these protein kinases phosphorylate one of the histone proteins, this particular one here, and partially reverses the silencing, so that all three classes of transcripts can be made: immediate, early, and late. So that ensures that VP16 is made, and that can start the transcriptional program. So it's that phosphorylation by this kinase, neuronal stress kinase, called JNK, which does the job. And then histone demethylases are made, which remove the rest of the repressive marks, and then you can get full transcription and eventually virus particle production. So that solves the conundrum of how you can get viruses without VP16. Very cool. I'll tell you that we don't have any understanding of this for any of the other herpes viruses. I'm going to tell you, unfortunately. It's very cool. Okay, next question. Persistence of herpes virus simplex in ganglia requires which of the following? Continuous episomal DNA replication, low-level production of virions, silencing of gene expression except LAT and microRNA, UV light stress or steroids, all of the above. Persistence requires which of the following? Okay, how did we do? Right. C is the right answer. Silencing of all gene expression except LATs and microRNA. There's no continuous DNA replication, right? There are neurons. You don't need to replicate the viral DNA. There's no virions produced during a latent infection. None. UV light stress or steroids reactivate. Nothing to do with persistence. So it's not all of the above. Next, herpes virus. So there's a family called herpesviridae, or herpes virus family, and it contains herpes simplex virus and Epstein-Barr virus. 95% of US adults are seropositive and carry the EBV genome in B cells. It's not a neurotropic virus. This is a B cell tropic. You get infected at an early age. You are often asymptomatic, but the virus can cause mononucleosis, which you have certainly heard about, and a variety of human cancers. Hodgkin's lymphoma, nasopharyngeal carcinoma, and Burkitt's lymphoma. So what's happening here is that, again, you are infected by saliva from your parents or someone you're kissing. You know, anybody who's not seropositive, by the time they get to college, they convert, they seroconvert. Virus infects these epithelial cells, reproduces in them, traverses them, and then infects B cells. It actually converts a B cell to a memory B cell. Why would it do that? Because a memory B cell lives forever, or a long time, right? Brilliant, brilliant. And That's where it resides latently, in the memory B cells. Then later on, at some stress, which we don't know what it is, a few latently infected B cells begin to activate their gene expression profile. They make virus particles, and those go through the epithelial sheet and infect someone else. So the B cell is essential for EBV latency, Epstein-Barr virus. The DNA is a self-replicating episome in B cells. A few viral genes are expressed, different from herpes simplex, but no virus particles, okay, just a few viral proteins. And the B cells, memory B cells, what they normally do is go to your bone marrow and lymphoid organs, spleen, lymph nodes, and it stays there. So this EBV is latent throughout your body. And these B cells are not killed by CTLs or antibody because they're hiding, they're not producing recognizable antigens. Only when reactivation occurs does some killing happen, but the reactivation is so quick that some virus-infected cells are going to escape. Okay, that's EBV. By the way, recently, in the past years, EBV has been clearly associated with MS. So people who have EBV, a fraction of them make antibodies against the viral protein, which cross-reacts with a neuronal protein, leading to multiple sclerosis. So people are working on a vaccine for EBV that would prevent, presumably, MS. It'd be great. All right, the third herpes virus, varicella-zoster virus. These are two different diseases caused by the same virus. Varicella is also known as chicken pox, and zoster is known as shingles. And so what happens here, this virus is infected by respiratory secretions and ends up in your upper tract, reproduces in primary lymph nodes, establishes a viremia, which spreads it to other organs. That makes a secondary viremia, which gives you the skin rash of chickenpox. I had chickenpox as a kid. I remember it very clearly. Very painful. Then the pox goes away, you recover, but the virus becomes latent in nerve ganglia that innervate the skin, not the trigeminal ganglia or that sort of thing, but these are, mostly they're in your trunk, and the virus remains in those nerve cells. And periodically they are reactivated to cause this rash called shingles. Typically you get it across your body here on one dermatome that's innervated by a particular ganglia. So obviously the virus has gone latent for many years. Shingles typically occurs in people over 50. So before we had a vaccine, we do have a vaccine right now, 99% of those got infected. 30% developed zoster, which would be 50 years, over 50 years. Two-thirds of them are over 50 years of age. Some young people develop zoster too, if you're immunocompromised or immunosuppressed. But most people, just as they age, they develop zoster. And the virus remains as an episome in neurons, non-replicating episome in neurons innervating your skin. But again, viral gene expression is like EBV. A couple of genes are produced, but no infectious virus particles are produced, and we don't know what triggers reactivation from neurons. In other words, you may get one or two shingles episodes in your life, so it's not like it's happening as frequently as herpes simplex 1, but we don't know what the trigger is for that. And I had chickenpox as a kid, so I had shingles, I don't know, in my 50s. I remember that well too. So there is a vaccine for chickenpox, but now there's also one for shingles. So people like me who got chickenpox naturally, you can be vaccinated against shingles and it'll prevent the emergence of that rash. and it's a glycoprotein vaccine. We'll talk about it later. Really good. Cytomegalovirus, the next herpes virus. Again, high seroprevalence. This is a chart of seroprevalence by antibody, by years of age. So you see the seroprevalence goes up. These are non-Hispanic whites, non-Hispanic blacks, and Mexican Americans. And you can see it's differing early on, but eventually converges at a very high seropositivity. This one is transmitted respiratory routes, virus and saliva. Again, your parents kissing you. Also, it's shed in urine, can be spread by sex as well. It replicates in white blood cells and in blood vessels, the endothelial cells of blood vessels. And primary infection, usually asymptomatic or a mono-like illness. You shed virus and saliva and urine for a long period of time. The infection is eventually resolved, but the virus becomes latent in myeloid cells in the bone marrow. So here's the myeloid cell. Here's a hematopoietic stem cell that gives rise to all the blood cells. And here's the common myeloid progenitor. So this is where CMV becomes latent. And then you can find it in monocytes and macrophages as a consequence. Okay, so CMV is a big problem in organ transplantation because to get an organ transplant, you need to be immunosuppressed, and that will start CMV replicating. And so a whole field of transplantation infectious diseases has emerged as transplantation becomes better and better to treat these infections, because everybody has CMV. So the assumption is it's going to replicate when you're immunosuppressed. The other problem is that the virus can cross the placenta, reproduce in the fetus, and cause congenital defects and death. And here are the numbers on the right. So if you have 1,000 pregnancies leading to live birth, 600 have had CMV before pregnancy. Most of those give birth to CMV negative babies. 6 give birth to CMV positive babies, and 1 to 2 will have permanent problems. And likewise, 400 women who don't have CMV before pregnancy, 7 will get it during pregnancy, 2 will give rise to CMV positive babies, and again, 1 to 2 will have permanent problems. So, this is hard because there's not much you can do with the population being almost entirely seropositive. All right, our last question is, what do persistent infections with EBV, VZV, and CMV have in common? A, B cells are essential for latent infection. B, may cause congenital birth defects. C, the viral DNA persists as an episome. D, the factors governing reactivation are well known. E, all of the above. What's in common? All right, let's see how we did. DNA persists as an episome, that's right. B cells are not essential, that's EBV. Congenital birth defects is CMV. The factors governing reactivation are well known. No, that's just herpes simplex. That leaves C. All right. Last virus, human herpes virus 6 and 7. These are agents of a rash disease in children called exanthum subitum, or sixth disease. There's 6 childhood rash diseases. Again, many adults are seropositive. Parent to child spread, just like the other herpes viruses. This virus infects lymphoid cells, endothelial cells, the liver, even the CNS and salivary cells, and goes latent, for HHV6 and monocytes and macrophages and hematopoietic progenitors, and HHV7 goes latent in CD4 positive lymphocytes. But the interesting thing here is that the viral genome in about 1%, or a certain fraction of people, the viral genome integrates into the telomeres of the chromosome. So here's the human chromosome. That's the telomere, the end. It's highly repeated. And this is the viral genome. It has telomeric repeats with it. And so this telomeric repeat will integrate into the host cell, telomeric repeat. So now the genome is at the end of the chromosome. And so about 1% of transmission occurs from parent to child via the germline, as if it were a retrovirus, right, a provirus. And so this is one strategy for latency to maintain the virus genome in the chromosome and for transmission to other hosts by the germline. I want to end with this slide, which really summarizes everything we've talked about today. This is the estimated burden of chronic viral infections in humans. So here are different viruses here on the X, and this is millions of people. So everybody has endogenous retroviruses, okay? So that bar is everybody. And then we have lots of viruses that are close. Anellovirus, HHV6 and 7, VZV, EBV, CMV, polyomaviruses, AAV, adeno-associated virus, parvovirus, simplex viruses, hepatitis B, papillomas, HCV, Kaposi's sarcoma herpes virus, and adenovirus, HIV, here's HIV, 35 million people. So almost everyone is infected with more than one virus. Why am I telling you this? So you want to figure out the cure of a disease. Let's take an easy one, diabetes. What's causing diabetes? Whatever you're going to do, every one of your patients is going to have a chronic viral infection. So you cannot tease that away for whatever you think causes diabetes. Well, maybe you could, but nobody does. People ignore this who are doing trials to figure out other diseases. And I think that's wrong, because obviously these can make a contribution in a variety of ways. So just think about that going forward, that whatever human disease you're interested in, a non-viral disease, there's always going to be a virus in the background which you have to tease out. Now, the Epstein-Barr virus, MS, story is very interesting. People always thought, how are we ever going to figure out the involvement of EBV and MS? Because everybody has EBV. What was the solution? Amazing solution. So when you go in the Army, the first thing they do is take blood from you here in the US. And they store it. And they take blood periodically. So an investigator got all these bloods, and it turns out that a certain fraction of people who go in the Army are EBV negative. And the ones who get MS later in life are the ones that turn seropositive. None of the negative people got MS. So you have to think creatively like that to be able to deal with having EBV in everyone. It's not impossible, but I don't know, people just don't think about it when they do their studies. Okay, next time we're going to talk about transformation in oncogenesis, how viruses are involved with cancers.